So hello, my name is Srecko Gajovic and I come from Croatian Institute for Brain Research, which is in Zagreb, Croatia. So I will a bit switch gears going to animal experiments. Actually, there is a common point of all of us that we want to solve the challenge of our society. So here it says actually the years of healthy eating and exercise have unfortunately added 5 more years to Melvin's life, because we have a good news and the bad news. The good news actually that we are going to live longer and longer. They say that a small girl born in Japan right now has 50% probability to reach 100 years old. The bad news is that this will not diminish the burden of diseases. Actually, half of the people older than 65 years have 3 or more chronic diseases. And actually we would have an older and older society, but we have to find a way as a society how to cope with these diseases. So of course one of the answers should be regenerative medicine. So according to this we got a project from EU FP7 project. This is REC-POTLAI Regional Potential, and the name of the project is GLOBRAIN. This is the acronym. To implement some research on mice for regenerative medicine, but our disease model is stroke, and we make the stroke in mice by this method, middle cerebral artery occlusion, which actually means that through the neck there is a very tiny filament which goes in the blood vessels of the brain, and when the filament stops, so it goes straight from the neck to here, and when it stops it blocks this artery, this middle cerebral artery, and as a result, this part of the brain will have a stroke. So the GLOBRAIN project has 3 main pillars. The first is to see what's going on if we want to design some kind of regenerative medicine for stroke, what's going on in the living animals. And what we want to design, how we want to help these animals, is by stem cells and combining the stem cells with biomaterials. So to do this, we actually arrange together several in vivo imaging modalities, meaning modalities that you can see what's going on in the living mice. So the first one is small animal MRI, then micro CT. MRI is of course magnetic resonance imaging, micro CT, computer tomography, and IVIS system. IVIS comes as an acronym of in vivo imaging system, but it's mainly based on light producing in the animals, as a bioluminescent imaging, fluorescent imaging, and very rare Cherenkov luminescence imaging. But I will go point by point, and all this will be arranged in some kind of facility, which is a basement which we are right now assembling. So here the scientists will come here they can work, but from here on this is restrictive access for the imaging, the MRI, micro-CT, bioluminescent imaging, and from here on again restrictive for the animals, which will be hosted here, mice, rats, and there are some, of course, rooms which are needed. And here one can grow the stem cells. Through this small window, the cells can pass to the animals where they can be operated. The animals can recover here, and then through the small window, they go for the imaging, and then they are imaged right here. So it's some kind of mouse clinic which we have designed in a relatively small space. So this is how the space looked like something like half a year ago. Then it turned to be better. But what is right now, the MRI is inside and it is just in the process of installation. So during this time, actually for MRI, we actually try to solve the question how to see the stem cells in the mouse brain. This question is, of course, to use magnetic nanoparticles. And one of our partners from Prague, Daniel Horak, actually produces for us the Magemite nanoparticles, which have been polyolizin coated. So we wanted to compare these custom-made nanoparticles to the commercial ones, and even on this light microscopy, you see the staining for iron that is much higher in this PLL. For PLL nanoparticles, the cells are neural stem cells. But to quantify all this, we actually had to install many procedures, of course, to say how many cells have been labeled, which we did with flow cytometry. Then to show that the cell proliferation was not affected at this concentration. So these are our particles. These are the commercial particles. So you see that actually the much lower concentration is needed to achieve the same results in sense of cell viability and cell labeling. But we have to prove that actually this goes inside the cells and not attached just outside the cells, which was done by electron microscopy. And finally, we had to show what is the mechanism of nanoparticles entry. This is cytochalasin D dependent, which means that some kind of actin dependent process is involved in this ingestion of these nanoparticles. About micro-CT. Micro-CT is not really for the brain. It is mostly for the bones. But we wanted to see, can micro-CT be used for imaging of stroke in small animals? And we solved this in ex vivo experiments, means one has to isolate the brain. Then this isolated brain should be some kind immersed in a contrast solution. These are contrast solutions which are normally used for human diagnostic purposes. They could be ionic or non-ionic. So these are the commercial names. And through this, actually, we decide to use this one, OmniPac, in a relatively higher concentration, but giving us quite a good noise to signal ratio and as well the good contrast. So then the animals have been operated to induce the stroke with MCAO. And at the first sight, it looks there is no difference, so CT cannot see. But when you use the software, actually you can delaminate where the stroke is, and the same brain can be then used for histology and as well stained for missile staining. And then when you compare the lesion volume and brain edema from histology and the micro-CT, you see that this is comparable, and actually one can use the micro-CT for these purposes. So going to this third imaging modality, IVIS, in vivo imaging system. So we mostly use bioluminescence imaging. The bioluminescence imaging means that you need to have transgenic animal. So here is how this machine looks like a big fridge. But actually what is inside, you open and you put the animal. Here is the CCD camera. And of course you need anesthesia that animals should sleep during the imaging. So sorry, this is in Croatian, but this is what I have. So what you need transgenic animals. So animals should have the luciferase gene. Luciferase is actually a gene from firefly, fly that goes around these summer nights and gives the light. So this gene was used, inserted in the mice. So when the luciferin as a substrate is given, then those cells having luciferase active, using ATP as an energy source for light, actually will then produce light in this mouse brain. And although the light is really tiny, it can go through the brain, through the skull, through the fur, to be seen by CCD camera. So how this looks like, of course, there are false colors according to the number of photons which go out from the brain. So this is the mouse head. The ears are in this sense covered, and the mouse has to breathe anesthetic. And here is the part of the stroke can be visible here. So we have several animals which have luciferase under different promoter controls. So one of the major is TLR2 promoter. So TLR2 luciferase mice means that we can follow the activity of TLR2 gene, which TLR2 actually is a gene generates a receptor, toll-like receptor 2, and this receptor can feel when the necrotic cells are around in the brain due to the stroke and initiate the reaction, which is mostly inflammatory reaction and activating the microglia. So you can see, as what one would expect, when you image these animals through the time, actually the peak is after the 2 days, but what was surprising that even after 3 months, there was significantly higher inflammation in this animal. So we actually wanted to see what's going on if we knock out TLR2. Was this be something to change the response to the stroke? And indeed, the inflammation, these are the microglia marker, the amount of microglia was lower. These microglia will proliferate less in these animals that are knockout, and there would be, of course, less activated microglia in these animals. But what would be the consequence of a stroke? And this was rather surprising, that when we look at the stroke after 3 days, the stroke was bigger in wild-type animals which had inflammation, which is more or less what one would expect, seeing general inflammation as bad. But in a sense of regeneration, when we looked at after 2 weeks, actually these animals which lacked the inflammation were worse, and the animals which had the inflammation were better. So this was confirmed as well on neurological score. So on living animals, you can score the number of, the maximum score is 18. You have 6 neurological tests. And in this context as well, wild types having inflammation have been better. So what is the mechanism? What we would suggest is actually that the apoptosis is delayed. So you see that actually after 3 days, there was less apoptosis in the knockout mice. But later on, the apoptosis is stronger and then continues on much longer than in the normal animals, which control this by some kind of inflammation. And seeing this reverse of the picture, first that the knockouts were worse and then better, but then worse, actually makes a question that many preclinical studies, when we test something, have been done only in one time point, which is normally 1 or 1 and 2 days. Only some clinical studies have been extended to longer time points. And we think that it is really important that after ischemia, the time is important if you want to challenge or to judge about your intervention or your therapy. So for this is another model, GAP43, and I will go relatively fast through it. GAP43 actually is a gene which helps the protein when it's expressed. It helps the outgrowth of axons. And in this sense, in adult animal, of course, it's very important during embryonic period, but in adult animal, it's some kind of repair and plasticity marker. And if you image these animals after the stroke, the peak of repair and plasticity would be around 1 week. But again, it continues quite long after the stroke in this chronic period, which is rather important for recovery of the mice. So we use this animal for in a very specific way, because we gave them some kind of a specific substrate which is commercially available, like VIVO Glow, but it is actually DEVD luciferin. So DEVD is a part of the protein which can be cleaved by caspase 3 and caspase 7. And when these caspase 3 and caspase 7 cleave it, then it's released to be able to give the light. So first, when we saw this, we thought, oh great, we will just inject this in the animal and image the light. We did not realize we need a luciferase mouse. So of course, we had a luciferase mouse. So if you have a luciferase mouse, let's say GAP43 luciferase mouse, then those cells expressing GAP43 luciferase and in the same time caspase 3 and 7, those will actually give the light. So actually you see a subset of GAP43 expressing mice which will have caspase 3 as some kind of apoptotic marker present. And this was already a bit confusing because GAP43 should be repair and plasticity, and caspase 3 will be apoptosis, but we had GAP43, so we tried, and we got the signal, which actually then we confirmed on histology. There is a correlation GAP43 and caspase 3 on the histological slides, and we actually show that ATF3 as a marker of stress is as well expressed here. So as a conclusion, actually both GAP43 and caspase 3 cannot be really taken as a marker of apoptosis or repair, but mostly as a marker of neuronal stress. And then actually, I guess the cell at one moment decides either to go to the apoptosis or will go for repair. So in this context, actually, I would suggest TLR2 can be used for biocompatibility testing, GAP43 for regeneration, and then this DEVD for neuronal stress. But how this relates to the stem cells and alginate, I will show, and biomaterials. I will actually show this on this example of combining neural stem cells and alginate as a potential for brain therapy after stroke in, of course, mice model. So we use neural stem cells from embryonic day 14 from fetal brain. We can culture them. We actually did many experiments without any biomaterials, but I will not show them. But what we showed that with 1% alginate solution, actually they live in vitro quite happily. So you actually use alginate, you mix with the cells, and then you push this in a medium containing calcium ions, and alginate will polymerize, the cells will be inside, and you see them here as a blue. And this made us to think, oh great, then we can combine them and put in the brain. So in vivo experiments, and we were not aware that actually translating this from in vitro to in vivo actually put us in a quite complicated position to actually speak about brain tissue engineering. Brain tissue engineering is complicated because first you don't have space, and second you cannot just put your combination in the brain. You need to solve how to put it in. So we always inject near the lesion, so in this penumbra region which we expect to recover. So this is a stroke, we inject nearby. But the amount you can inject is very small, it's actually 1 microliter. And then actually in sense of alginate, you have to be sure that alginate will polymerize inside. And we use for this solution which really slowly release calcium to get to polymerize this. Then we can, of course, follow the stem cells because they are marked with fluorescent markers. And finally, we could test, is this biocompatible with our TLR2 luciferin mouse? So you can see the positive control is LPS, so a lot of inflammation. Another positive control, MCAO, stroke, lot of inflammation. The negative control is PBS. Even in PBS injection, you will get some inflammation in the brain. But when you inject alginate, and as well alginate combined with the cells, actually it will be more or less comparable with PBS, which means the whole procedure is biocompatible. So in some sense, this combination of biomaterials and stem cells is brain tissue engineering. Of course, maybe on a very basic level. We have more tissue engineering in peripheral nervous system or in spinal cord injury. But indeed, the brain tissue engineering seems to be a rather peculiar way. And I put this on the map because I want to actually, I think that there are more people, more groups doing this, but what we have now is only Croatia, where I'm from, doing brain tissue engineering. So it would be very nice if you would be the first, but I think that this map should be more red. And if you know someone, just let me know, or just Google low brain if you forget my email address. Here are the acknowledgements. And this is the Croatian Institute for Brain Research where all of this was done. Thank you very much.